once again, Jonathan Spyros, thank you so much for finally, arranging to talk to us, because, I mean, you have been involved in some of the most interesting research. You had some of the most exciting PhDs probably of anybody's careers. And, it's really interesting also to get some of your thoughts about how you feel your work progressed. Spyros, congratulations on your recent PhD defense, which was successful. You had a very fancy hat. I saw.
What do they say? May you live in interesting times, and it has been for virology PhDs, hasn't it? The last few years. So
there was more sparkling wine and less talking after
yeah, I can imagine. So today we want to talk about a new preprint that you uploaded on an archive. So this is not peer reviewed yet, but it will be submitted, or it is already submitted, and it will come out at some point in the future. And the preprint is called "The Recency and Geographical Origins of the Bad Viruses Ancestral to SARS-CoV and SARS-CoV-2." So this is a bit of a mouthful of a title, but basically it means that you looked at bad viruses and tried to figure out what it means for the origin, not only of SARS-CoV-2, but also SARS-1. And, I'm happy that Sam is also with us, because we have to break down a few key biological issues here a little bit to really lead people along the way to understand. And the first one of these issues is, what is viral recombination? How does it work? Is it that every virus does recombination? Can you please explain just, to a wide audience, what is recombination?
Is this my cue, Jonathan? Is that
probably?
Go on. We'll work from left to right. We'll keep it. There we go. Spyros, you lead us off.
So recombination is an interesting phenomenon that actually most viruses do, especially RNA viruses. Some of them do it more, some of them do it less, and coronaviruses in particular do it quite a lot. So recombination is when, imagine you have two viruses. They have their own genomes. They're somewhat distinct to one another. And then when they find themselves in the exact same host, in the exact same cell during a co-infection, they replicate as a single virus would do. But essentially the polymerase that replicates the genome kind of skips a bit and ends up in the other virus's genome template, which means that the final product will be a recombinant, a chimera, if you like, of the two co-infecting parental virus strains.
So you have basically two prints of something, and then you have the replication machinery that runs and runs, and then decides to jump to another one and finish up, or and jump back sometimes and go on. Is that how you imagine it?
Yep, that's right. So biology and biochemistry and physics and all the natural sciences are quite stochastic, and that's how I like to think of recombination as well. And in fact, a lot of the recombinants will not be viable. It'll be like wrong jumps into the wrong place, incomplete genomes. But the recombinants that we sample in the wild is viruses that recombined and then were successful to transmit further on.
Does that suggest that the level of recombination, or the rate of recombination, is much higher than we actually see, because we only see the ones that go on to replicate?
Yeah, we tend not to estimate rates of recombination from sample genomes exactly because of that. So to calculate or estimate the real rate of recombination, you'd probably need to do in vitro experiments to figure it out. So you can't really calculate the real.
But this is interesting. So it could mean both, that either these viruses allow for more recombination, or they are more tolerant for it, and can create a lot of offspring that is still viable, although they have a lot of different pieces put in left and right, and still somehow remain viable. Right.
And apart from the cellular level, we have to think about the ecological level, right? So this means that the amount of recombination we'll talk about, and we see in these SARS-related coronaviruses circulating in bats in China and Southeast Asia, means that they definitely infect the same hosts, the same populations of bats, and tend to find themselves in co-infections in the same individuals and cells.
The only reason I flag this up is because certain of our friends who shall remain nameless in this discussion seem to suggest that this really doesn't happen that much and we shouldn't really worry about it, particularly when doing calculations of, you know, ancestors and these kind of things. We should really be looking more at point mutations rather than recombination. But this seems to be something that happens all the time with the viruses that we know and the coronaviruses that we look at when we're thinking about the ancestors of SARS-CoV-2. Is that fair?
That is very true. Yeah. So we've looked at SARS-related coronaviruses. We've worked with Darren Martin's group in the past, exploring recombination in all different subgenera of coronaviruses, and we see the same pattern everywhere: excessive recombination. These viruses really like to do this.
So this is good. Jonathan, before we come to your specialty, I have another question, maybe more towards Spyros. So when you find a genome and you don't know, how do you know that it's recombinant? You know, it's just for you, it just looks like it's a new virus, is functional. How do we even find out, oh, this is a recombinant? Any suggestion? Is this just totally obvious, or do we need to sample a lot of bad viruses to make these estimations? What is your feeling?
Is that for me or Spyros?
So it's for Spyros. I said before we come to you, Jonathan.
Oh, before,
okay.
Sorry. I was waiting for Jonathan to answer. Sorry.
Don't worry. I can cut any bits that like are slow. Don't worry.
Sorry, it's been a long day. Right. So the way we figure out what is recombinant and what is not is mainly by comparing each segment of a potential recombinant genome to other genomes that could be the true parents or of the parental lineage that these viruses came from. So usually all these algorithms work in triplets. So you consider 3 virus genomes, and then you kind of say across the genome, is there any bits that look more like virus 2 than parent virus 2 than parent virus 1? There's other ways of assessing recombination. For example, in the paper we're discussing now, we use a slightly different approach. We don't actually determine which viruses are recombinant, but we use an algorithm to chop up the genomes into segments that will give you distinct evolutionary histories, which means that if you put all the genomes in the right place, and then chop up the genome in segments, what we refer to as non-recombinant regions in the paper, and then the non-recombinant region 1 will have one evolutionary history, and the one next to it, non-recombinant region 2, will have an evolutionary history that's different from that of 1.
So this is very complicated, and we will spend probably the next 10 minutes going a bit into the details. But just in general, Jonathan, and now it's time for you. Usually when we have a viral outbreak and the virus starts from an introduction and starts moving, we kind of can figure out their timings in a certain way. It's called phylogeny. Can you explain what is the idea roughly behind how can we measure the time, and then how can we calculate the evolutionary history kind of back into the other way from this outbreak? What is the rough idea behind it?
I mean, so there we're going to take all the given different genomes that we have, and we start comparing how similar they are to each other and building what we call a phylogenetic tree. It's essentially like a family history or family tree of these viruses or of those segments of the viruses, which we'll go into more detail, I'm sure, as we talk about this paper. And as we start reconstructing that family history, we can have a better idea of when ancestors circulated, when a given virus might have been introduced into a population, and how long that virus might have been circulating in a given population as well.
But how exactly does this work? How do you know, for example, that, they are like, I don't know, 7 mutations apart. This means it was like, a month ago that the most recent common ancestor of them or last shared ancestor of them was introduced.
Right. I mean, so when we start building those trees, what we want to do is calibrate them with time. And so we incorporate the temporal information into our tree building. And that way, when we can start building those connections, say, like you have 2 that are 7 mutations apart, and we can sort of infer what the evolutionary rate is between these viruses over time, we can then start figuring out how long, let's say, each branch of the family tree is, when that ancestor might have circulated, and so on. And so it's the combination of the genetic differences and that temporal information when we sampled them.
So the temporal information is really like, the better the surveillance is, the better is the temporal information, right? For when you have, I don't know, a new virus outbreak, and then you see, the first patients roughly were here. Also with epidemiology, you match them, let's say in December, and then in January, you have a different set of genomes, and in February, you have a different set of genomes. And this is how you timestamp them. And then, but the tree building itself is with the temporal information, or is the tree really based on the distance, on the time distance, or on the mutational distance, or is it both?
It's both. You can build trees without the temporal information, resulting in what we call like a mutation tree. And that's typically done, at least today, like under a maximum likelihood framework. Or you can start incorporating the time information as well, in which case it would be time and genetic distance. And the time adds another component in which we can better resolve internal parts of the tree.
So it's basically extra information. So whenever we have good timing estimates, it helps us a lot with really narrowing down or refining the tree. Is that fair to say?
Exactly. We're trying to figure out how likely a given tree is based on the number of substitutions we see, based on the timing, and so on. And the better that temporal information, the more refined that temporal information, the better our tree can likely be.
Okay, well, so I will summarize what I so far understood. So coronaviruses, they make a lot of recombination, and that means that when you look at the parts that are or the parts of the genome, you see some of them shift a lot on the recombination breakpoints, and other parts, they are kind of stable. And for each part that is stable, it's kind of it has its own little evolutionary history that comes from the past. And then they all kind of come together to form the virus. And if you know the history of all the parts, hopefully you know a bit more about the closest ancestor of SARS-CoV-2. This is roughly where the paper is going to go. Good. I think
this happens a lot. So we shouldn't be, when we're thinking about the history of a virus, we shouldn't be thinking about one mutation, one mutation, one mutation, one mutation. We should be thinking about these separate pieces. And
we should think about where do all the puzzle pieces come from this genome, right?
Yep. Essentially, it all comes down to the mutations, but if recombination has taken place, then the mutations are cheating.
Yeah, you have to factor in all these different components when you talk about it. You can't just rely on one.
Because there seem to be a hell of a lot of papers out there that do naive kind of just point mutation analyses and then say this couldn't have happened, this couldn't have happened, there's no way that this could have happened. But if you don't include this recombination, particularly with coronaviruses, it just seems that your paper is almost meaningless. Is that fair?
As long as there has been recombination, but it's very important to check for recombination, and at the scale we're talking about with these viruses, and specifically about these viruses, accounting for recombination is crucial.
So next question then is kind of logical. Where exactly get these viruses the opportunity to recombine? Do you know what is known about this? Do you have any insights to, why do we have this strong recombination patterns, and where does it happen in the world?
Let's go to Spyros, because otherwise we're going to get another awkward pause.
Thank you.
All right, I'm going to dive in.
So where does recombination happen? Recombination has to happen in the bats. It comes back to what I previously said about the viruses co-circulate in the same bat populations. I mean, we see sampling efforts that have gone in a single cave and sample diverse viruses that show recombination between them from like bats living right next to each other. The best example, it's really difficult to kind of observe that in action in the bat populations, but we've basically seen it with SARS-CoV-2, and we've monitored it extremely well. It's to me the best proof of concept of recombination happening in a population that's constantly infected by distinct strains of a given virus. So the currently circulating lineage of SARS-CoV-2, XBB.1, whatever the alias is now, is a recombinant. And
a recombinant between different Omicron variants?
Yes, and we've observed recombination between earlier variants. We've seen recombination between completely distinct variants of concern. We've seen recombination between Omicron and Delta. But the prerequisite for recombination to happen is that both distinct strains, parental strains, co-circulate. So we only saw kind of confident Omicron Delta recombinants in between the time where Delta, kind of the end of the Delta wave and the start of the Omicron wave, where there was enough co-circulation of these two strains.
So we really, unfortunately, had a good host population that was well surveyed, which was us humans, and we saw different variants emerge and then also sometimes recombine. And as you say, the more recombination we have, this can have evolutionary beneficial or evading immune system beneficial traits. And this is what we see today within the Omicron variants, but also before, of course, we saw this a little bit.
Now, one thing I would add, also though, is that even before then, it's very possible there was recombination too. You need distinct strains, but they don't actually have to be different or very different. They could have been close to identical or identical strains within the same person, and then you just wouldn't pick up recombination, right? And so that it's very possible that happened, and maybe there was onward transmission, but we wouldn't know.
Yeah, the higher the diversity of SARS-CoV-2 variants, the more likely we will see the recombination, because if you have some very similar SARS-CoV-2 viruses that only differ, then if they recombine with each other, we wouldn't really see that, I guess. We only see it if there's a large chunk from Omicron with Delta, for example. I think this is one that's very clear if you would see that.
And diversity is key. And when we're talking about SARS-CoV-2 and then the SARS-related coronaviruses, we expect that there's a lot more diversity circulating in the bats that have been the reservoir hosts for thousands, hundred thousands of years, compared to humans, which have been the SARS-CoV-2 reservoir hosts for what is now 4 years ish.
So I was just gonna ask that there, Jonathan. So does that mean when you're doing the analysis and you're looking for these unique non-recombinant sections, does that mean that you probably are making an underestimate of how many distinct sections there are? Because there's
I would say it's typically an underestimate of the number of sections that there are. And in our case, and Spyros can speak to this more, we're not necessarily trying to capture every single recombination event or every single non-recombinant region. We're trying to look for each distinct evolutionary history among the non-recombinant regions. Because if you have two adjacent regions with an identical evolutionary history, whether they get combined into one segment or not, it matters a little bit, but it's not that substantive.
Yeah, makes sense.
I have one more question with this recombination breakpoints, because you've seen also, I think, between the comparison SARS-CoV-2 and SARS-CoV-1, that sometimes these breakpoints are very close together between these two different viruses. Does that mean that the area of diversity, let's put it, is, separating distinct genetic elements between the virus? With the spike, we know, for example, this can happen surrounding the spike region, surrounding S1, S2, stuff like this. Can you speak a bit more about this?
In terms of, so you do, so there's different kind of layers to this. we do not, because we're inferring, so to infer an evolutionary history out of the non-recombinant regions, we need enough mutational information within it. So we reach this trade-off where we would get rid of, we would certainly get rid of all recombination if we separated the alignment into single nucleotides. There wouldn't be any more recombination. But that's completely uninformative, right?
Yeah.
So we're reaching this trade-off where we want to find
informative segments.
Exactly. That have kind of enough information, that break up the alignment into bits that have enough information within them to infer the evolutionary histories from that information. So in our analysis, we actually have kind of a threshold of 100 nucleotides, where then we kind of combine the inferred breakpoints. And the algorithm we're using is also quite conservative with nearby positions. However, we still do see kind of more breakpoints around known hotspots that in previous analysis that was actually looking at this kind of thing.
Yeah, because the segments are not all the same lengths, right? You have quite a variation between, let's say, 300 nucleotides up to 2,500. So it clearly means that some longer regions are a bit more stable, and some shorter ones, they're a bit, hotspots, like you say, they like to switch out. Is this because of RNA secondary structure? Is this of selection pressures, or a combination of both? It's very intricate, probably.
Yeah, that's a very good question that I don't. I only have a hand wavy answer about. A lot of the things. So for spike, we think that some of it is selective. So we see more viruses that have recombined their spike because they're actually better off in a naive, in an immunologically naive and genetically naive population. However, the viruses could have developed, because this is beneficial for them, could have developed mechanistic reasons why they're recombining at these specific places. So it's very difficult to distinguish between selection and mechanistic mutational recombination, both because it's extremely difficult to measure and because they might not be distinguishable per se.
And because we probably still are largely undersampling viruses, and we don't have the perfect history, because some of these viruses are very old. And so now the next question goes a bit towards maybe Jonathan again, is, how far can you go back in time when you have samples? Let's say, you sampled back maybe the first viruses with genomes, maybe 10 years, 15 years, something like that. How can you ever go back? Because there were also papers, I think, from you claiming, this is, maybe 50 years back or something like this. How far can we go, basically? Does it depend on the length of the segment? On what does it depend? How far we can go back in time?
So I think there's two questions in there.
Perfect.
I'll answer the second one first, where you asked, does it depend on the length of the segment?
Yeah.
The length of the segment is certainly important, right? The shorter your segment, especially if it's below a certain length, the less phylogenetic kind of confidence you can have about.
If you took that down to the limit, it would be like nothing, right? If you had one, it would just
right. If you take it down to one, you're very uncertain, and the longer you get, the more confident you are in your estimates, right? At pretty much each internal node of the tree. Once you reach a few hundred, you can start becoming decently confident, and we allude to that in our paper. If you have a few thousand, that's even better, of course, but we don't always get that when we're doing our recombination analyses. So that's the first question. The second question, in terms of how far we can go back in time, there has now been a sizable body of work trying to infer deep history of viruses, including coronaviruses. And some people have different ideas on how far back we can go in time, but I think we're able to consistently show that for, let's say, the SARS-CoV-1 like viruses or the SARS-CoV-2 like viruses, we're going back thousands to tens of thousands of years at least. if we're talking SARS-like viruses in general, we're now going into hundreds of thousands of years, if not millions. And then coronaviruses generally, we're already like certainly in hundreds of thousands, and then most likely millions of years. We're able to make those inferences through a variety of techniques that basically try to understand that you can have multiple substitutions at the same site. And when you start accounting for that, you basically see that it's actually been evolving and circulating longer than you otherwise would have estimated. It's that key kind of inference point that we need to do this.
Because when you have a certain position, then you can have, a mutation and then a reversion or a mutation into something else. And this you have to also consider at some point that, there's a progression of mutations on the same spot, right? So it seems very complicated to me as an outsider, probably very difficult. So what do you really need to kind of, go that far back? What do you need? More genomes? Do you need better methods, better statistics, or a combination of these, or more knowledge?
You certainly need the methods, right? And there are different methods looking purely at selection, and then there are methods that try to be a bit more what I think some of us would call mechanistic, and are actually trying to infer the likelihood of having multiple substitutions at a given site. So like one of our co-authors, Mohan Ghaffari, has been really kind of leading the development of one of those methods. And it is like through his involvement that we've been able to make these deeper inferences. More genomes helps too, because the better understanding you have of the diversity, the better understanding you'll have of how deep in time that history and diversity goes.
All right. So now we have a lot of different pieces of information. And now I really want to know, what can be done with that type of information? What did you do in your paper? Because you created some kind of, a simulated recombinant ancestor of SARS-CoV-2 by putting all the different histories from all these different non-recombinant segments together. So why did you do it? Well, what can we learn from doing this kind of exercise?
Spyros,
anybody?
Tell us.
Yeah.
I guess the point here is trying to figure out how close we can get to the direct ancestor of SARS-CoV-1 or SARS-CoV-2, any virus of interest. We could be doing this for a random bat virus if we're interested in that, but obviously the human viruses are of interest here, and trying to get as close as we can to that, both in terms of dating that Jonathan just spoke about, and in terms of sequence, right? And what we call it in the paper is the closest kind of inferred ancestor, because we're restricted by what we know is out there, right? And a lot of the things I think Jonathan can kind of chip into that. The argument that because there's so much recombination, you can keep sampling, and the more you sample, the higher the overall recombination unaware pairwise identity you've got to new viruses you've sampled compared to your virus.
So I did not understand this. Can you please? Why is it that when you sample more, it gets it kind of you get more accurate, or I guess the error margins go down or something like that? Can you please explain this again?
So first of all, you get closer just by sampling more. Just because the more you sample, the more viruses you have, so the more chances that you will sample something closer to what you already have. And that's just comparing without inferring any the recombinant common ancestors or any simulated kind of sequences. So the actual genomes of the things you sample are closer to what you are looking for. So in the paper, we mentioned that the overall genome similarity closest relative to SARS-CoV-1, which emerged in late 2002, was actually sampled in 2020. And that's just because now we've sampled a lot more viruses that are out there. Now, apart from the overall genome similarity, if you account for the recombination, instead of saying I'm looking for the full genome similarity of everything that we keep adding, you say I am looking at all the recombination events and the how, including thinking about recombination, how the similarity increases. So instead of getting one genome at a time, you mix them up and just get the closest bit across the genome for each new genome that you sample out there. So that means that even though by sampling, we get closer to an overall genome that's out there, to similarity to a human coronavirus, if you account for the recombination, you get even closer, because you're looking for bits. There's so much recombination that you're not looking for a full genome related to the.
You're looking for floating genetic elements at this point, right?
Yeah.
Because you're saying basically, okay, there's viruses, and sometimes maybe you get lucky and you find a virus that's overall very close. But even if you find viruses that are not very close overall, they might be close in one specific segment.
Yeah.
And this already helps your information. Because then if this
bits of genome that are out there. Yeah.
Jonathan?
I mean, I fully agree with everything Spyros said. So I do think it's also interesting, though, where more sampling, like by definition, is going to give you the opportunity to sample more closely related viruses. But sampling in and of itself isn't the answer, right? You actually do need those viruses that are more similar. So for instance, in our paper, we have roughly 141 or 140 SARS-CoV-1-like viruses and 27 SARS-CoV-2-like viruses in our separate data sets.
Yeah.
But you'll see that for when we infer our recombinant common ancestor, which is essentially the genome or the sequence for that closest inferred ancestor across every non-recombinant region, stitched together separately for SARS-CoV-1 and SARS-CoV-2, they're actually pretty similar. They're both around 98.6 to 98.8% identical to their respective SARS-CoV, whether SARS-CoV-1 or SARS-CoV-2. And so despite the fact that there are roughly 5 times as many SARS-CoV-1-like viruses than SARS-CoV-2-like viruses, that doesn't necessarily mean you're going to have a more closely related recombinant common ancestor.
Mm-hmm. This is really fascinating, and I want to talk about this a bit more, because I remember, back in the day when Shisheng Li found the origin of SARS-1 in the bat cave, that close virus ancestor. She also did not find, one that was too close, but she said all the genetic elements are in that cave that she sampled there in Yunnan. And this was good enough, but this was still, I think, below what actually you stitched together now. From SARS-CoV-1. So in this case, we still, although it was already clear that it came from bat, this approach really brings that even closer to the ancestor, not only of SARS-CoV-2, but also SARS-CoV-1 and the geographic region. We're going to talk about this dispersion a little bit later. I just have a quick
I think
question.
We've had some... Yeah. Oh, sorry, go ahead.
No, please go ahead.
Oh, no, we've had a whole series of very fascinating, insightful papers on SARS-CoV-2 and its origins. And when we had the initial outbreak of SARS-CoV or SARS-CoV-1, however you call it, there were a number of papers like that too. But now that we have, more refined techniques, more sampling, and so on, it's really worthwhile to go in and try to once again re-understand, re-infer all of these histories of SARS-CoV-1, because then we'll probably be able to get a better understanding and resolution of that history.
Yeah.
So I just wanted to jump in, Philip, before you go into the next question. So this inferred common answer, for example, for SARS-CoV-2. So we've seen a couple of articles, and these articles seem to get people really worked up, with virologists saying you will likely never find this particular.
We'll not find bat zero, basically.
Yeah, you will not find a bat zero. You'll not find the ancestor of SARS-CoV-2 in a bat, but we might find all of the puzzle pieces throughout throughout bats. Do you want to tell us a little bit about that, Jonathan? Why that might be the case, that you wouldn't expect to find that, you call it Rexia in a bat somewhere, or how unlikely it is to find that virus somewhere in a bat whole?
Sure. I mean, well, first, Vexia is inferred, right? It is not an actually sampled thing.
Yeah.
So you're obviously not going to find that.
Yeah.
And it's only inferred based on our current sampling, which by definition, and of course, is very subsampled.
Incomplete, yeah.
But more to the point is that, so that Rexia inferred to whatever time frame you want it to be, has since both evolved and recombined, right? So since 2019, we've seen more evolution and more recombination, or rather, we might not have seen it, but whatever viruses have been circulating in bats have just continued to do so. And so whatever we sample now is going to be likely a more mutated and more recombined version of what was in the past. And so we're never going to actually sample any sort of predecessor because by definition, it no longer exists. Yeah. It's very possible that it also swept through a population and burnt out, and everything that we have might be a little bit more distant. It's also possible it kept circulating and just kept evolving and recombining, right? We won't know unless we just keep sampling and trying to better understand these histories. But what we are looking for isn't just puzzle pieces, or rather isn't specifically puzzle pieces that compose some sort of ancestor. It's puzzle pieces that descend from the puzzle pieces of that ancestor.
Yeah. And then are closer to what we see in the,
and then we can, even though they might have recombined and evolved, once we have them, we can once again infer their deeper history, because that we can infer that they are descended from that given ancestor.
Yeah.
This is actually the perfect introduction for my next question, which is, how do you see anybody faking this genome and its evolutionary history in a lab? They would have to fake 27 different evolutionary histories. Is there any chance this is possible, that this virus was created, in the minds of somebody in silico, or taking existing viruses at the time, Reggie 13, and just, inferred this genome and then printed it out? Is there any chance that this is not the natural virus? In that sense, from its evolutionary history,
because we've seen this a lot. The idea that you could introduce a couple of viruses here and there.
You copy paste them together basically.
After the start of the pandemic, and that has completely fooled your recombination analysis, because they've now given you the puzzle pieces that you need. How do we push back against that possibility?
I mean,
do we need to? Sounds like we don't, but
it's when SARS-CoV-2 the first genome was sampled and people were looking at it, right? We really didn't know of that many SARS-CoV-2 like viruses, because that lineage wasn't that interesting. There were some published recombinants that included kind of bits that were SARS-CoV-2 like, and a lot of the fully SARS-CoV-2 like viruses were sampled after the pandemic, because sampling efforts in more countries and more diverse environments increased, or had already been sampled. They were stuck in freezers, and no one had sequenced them because people didn't find that interesting, and everyone's very busy. Now that we have all these SARS-CoV-2 like viruses that we include in our methods and our efforts to understand where this virus came from, a made up kind of genome, SARS-CoV-2 being a made up genome, is absolutely nonsensical. It's bollocks. I mean.
So it's impossible that somebody had the idea of, I take, a couple of viruses and I have a new bad viruses found, basically, and then I stitch together, because it didn't have the right spike, it didn't have the right, let's say furin cleavage site or whatever, and I just stitched it together, and now it looks like these 27 pieces, they just came together to make it appear that there's this different evolutionary histories. It's unfakeable. Technically, right? I would say there's no way one can fake
there's
this.
nuance to the arguments, and that's where kind of grifters grasp onto, right? But for example, the furin cleavage side, we know of SARS-related coronaviruses that have kind of similar sequences that look like predecessors of the furin cleavage side that SARS-CoV-2 has, but we don't have a SARS-related coronavirus with this exact furin cleavage site sequence. But overall, it's, and then you can go into impossible and improbable differences.
Don't use these words anymore. They are taboo.
Yeah.
Otherwise, you're front of Congress sooner than you think.
Yeah, they've been banned.
Yeah, Jonathan, be careful what you say. I'm not a US citizen, so
yeah, Jonathan, you have to be.
Just leave it at no. I just leave it at no. I wasn't going to add anything.
All right, so it's a natural virus. We established that, but this natural virus could still have, been sampled by somebody, out there in the world, technically, right? Just to keep it formally on the table. Good, but now I think the paper gets more interesting and also more daring, I would say, because you did something very interesting with the geography. And I don't know who wants to take this question. What exactly did you do with the geography and the timing, and what were your findings there?
Jonathan, let's go to you for that question.
Sure. So after we did our combination analysis and we inferred the evolutionary history of each given component, we were interested in two things. The first part of the paper looks into the timing of the closest inferred ancestor of both SARS-CoV-1 and SARS-CoV-2. This is basically the node on the phylogenetic tree that is directly ancestral or directly above or parental to either SARS-CoV-1 or SARS-CoV-2.
This is where all the trouble started, basically. This is leading forward from there.
I mean, it is, based on current estimates, kind of the farthest point that we would look at, right? And everything else would be more recent.
Okay.
And what we're interested in is actually the closest inferred ancestor that is most recent in time.
Yeah.
And we find that for several regions, for SARS-CoV-2, we infer a close inferred ancestor that circulated only several years prior to the emergence of SARS-CoV-2. For SARS-CoV-1, we actually infer closest inferred ancestors that are even more recent, only 1 to 2 years before the emergence of SARS-CoV-1.
So I have to stop it for a second to just digest it. So we are looking basically of what was the most likely virus in an animal reservoir or something before emergence in humans, before we first noticed it in humans. And from that, we kind of so this is kind of, we want to know, when did this virus basically exist and how much did it take once this virus existed until it spilled over into humans, basically. And then, it could be that, it's been circulating for decades and then suddenly it spilled over, or it could be all the recombination pieces came together and once they were together, suddenly it would erupt into humans. So is that a fair summary? And what is your feeling behind this?
Generally, but I would add two pieces. One, not animals generally, but bats. We're specifically interested in these viruses circulating in bats and their evolutionary history in bats. And then two, we're also doing this because many people will, as you alluded to earlier, naively do whole genome comparisons and say, SARS-CoV-2 and this other genome, let's say RAG13, were decades apart. But then once you start looking at individual pieces, you see, well, it actually, the stuff that's most similar to SARS-CoV-2 was circulating very recently, not decades ago necessarily. And that's the interesting component of the timing question for us.
This is really interesting because this could also mean that, either, maybe we increased, recombination of these pieces by, invading ecosystems, getting bats under stress to recombine more or shed more or something like this. Or it could just mean that, suddenly, we these viruses, they popped into existence, and we were unlucky that, once a dangerous virus like this popped into existence, before it mutated away again, it was dangerous to humans and it spilled over.
And
I don't. Oh, sorry. Go ahead, Spyros.
Oh, sorry. I was just going to say that there's many more layers to this. For example, something that we don't really touch on in our study, but ACE2 usage. So the vast majority of SARS-related coronavirus sampled in the wild cannot bind to human ACE2 at all, or bat ACE2. So we don't really know what they're binding into and whether they could potentially be a threat for human infection. But this SARS-CoV-1 like viruses and SARS-CoV-2 like viruses, the kind of closest ones in the spike, both have the ability to bind to human ACE2. So we're really zooming into the populations of these viruses that already had the ability to infect humans in the first place.
I see.
And also, it's like, you were mentioning how we maybe got unlucky in terms of this virus popping into existence. I actually don't necessarily think that's the case. I think there are probably plenty of these viruses circulating around. What we got unlucky with is how it emerged, right? When and where it spilled over, and the conditions that were ripe for extreme spread of such a virus, as opposed to the fact of this virus existing in and of itself.
The delivery of that virus into a situation where the chains of transmission could begin.
I mean, Jonathan was also leading on other studies that we know about, but maybe not everybody who listens in. So in 2021, for example, you did simulations that showed basically that if this virus, broke out somewhere in a remote village, the likelihood of onward transmission was way lower versus how it happened. It needed kind of to be a big city with a lot of hosts and a lot of opportunity for the virus to jump over. Maybe you spent a few more sentences on this, because I think it's a very important finding.
That's exactly right. So when we talk about, let's say, a given market or any location being the emergence point of this virus, that doesn't necessarily mean that was absolutely the first time this virus spilled over into humans, right? If you have a long, let's say, supply chain with animals and opportunities for it to spill over, maybe it did spill over, right, much earlier, or let's say months earlier or something. But maybe the conditions there were not right for subsequent transmission and I don't want to use the word endemicity, but just like being able to like sustain itself in a human population. And so you needed a very densely connected network, essentially, in this case, a city, that would provide the right conditions for SARS-CoV-2 or SARS-CoV-1 to sustain itself further.
So I want to talk a little bit later about the conclusion of how you think it made it to the city. But first, we still have to talk about the geography, because I think this is very critical as well.
So why don't I pass that over to Spyros? I just talked a bunch.
Right, geography. So remind me again exactly what you'd like me to talk about.
So you showed in your paper basically that you can narrow down the geographic regions where the origin, the ancestor of SARS-CoV-2, the one that spilled over, most likely circulated. And this has to do basically, you would think bats, they have a huge region all over Southeast Asia, but then you narrow it down quite a bit. And how did you do that, and how solid is that?
Right.
You got really excited there for a minute. That was nice. I enjoyed that.
So it's like trying to zoom in. So we're thinking about trees, and then the maps come afterwards, right?
Yeah.
So we have a tree. We know which nodes in the tree, which tips in the tree, and internal nodes, common ancestors between viruses, are the closest to SARS-CoV-1 or SARS-CoV-2. And then since we have sampling locations for each one of the tips in the tree, we can literally project the tree onto a map and do a bunch of maths that other of our collaborators like Simone Delicourt and Philippe Lemay would be much better at explaining them. But essentially, you zoom in the parts of the map that correspond to the parts of the tree closest to SARS-CoV-1 or SARS-CoV-2. And we already said that we split up the genomes into non-recombinant regions. And for this analysis to kind of zoom in, we pull all our non-recombinant regions together, all our inferences, phylogeographic inferences across non-recombinant regions. And we can infer the kind of best estimate, kind of spatial estimate. And another novelty of the methods we use in this paper is that, so phylogeography, it's usually easier to do discrete phylogeography, which means you have, I don't know, like Vietnam and Laos, and if you have two viruses from Vietnam and one from Laos, then the node connecting them would be from Vietnam or whatever. Here we have actual coordinates. We're doing a continuous diffusion across the map, and we can essentially plot this blob that corresponds to the most likely location based on all the information with kind of clusters.
I mean, this seems really complicated. Is it really that you basically overlay for each piece kind of a geographic kind of, density map of for each, diversity, each piece, and how big it's kind of the beds where it was sampled? And then if you have all these different diversity, these different blobs together, you have some kind of a distribution that goes up on the map. Is that how you imagine it, basically?
Kind of, yeah. That's basically it. We could have done. I'm sure if we had some better like animation training, we could do some fancy animation. We resort to maps produced by tedious R code, but here we are. And then essentially, once we have those hotspots, what they represent is based on the sampling we have. Where is the location of the node in the tree connecting SARS-CoV-1 or SARS-CoV-2 to its closest known bat, SARS-related coronavirus?
And I guess, is there an assumption in there that bats kind of just slowly kind of diffuse around, fly around, and move quite slowly from their colonies? Like, because if you've got big lumpy movements, that's sort of going to break down, I guess. But I guess they stay vaguely within their ranges. Is that an assumption that you have to make there?
There's no assumption in the rate of diffusion.
Okay.
However, we assume the process of diffusion.
Yes.
And we
from a
tried
point,
dispersal as well. And I guess the results are quite similar. We believe that diffusion is a slightly better way to estimate things. I don't know if Jonathan wants to add anything to that.
Because I imagine you can't rule out somebody went and grabbed 1,000 bats and just moved them up to the north of the country or something, right? Nobody's probably going to do that, but I guess it's something that somebody could push back with. Jonathan?
Yeah, so I guess I would add a few things. So kind of to bring what Spyros was talking about and what I was talking about together, when in terms of the points where we think that ancestor circulated and the time that we think that ancestor circulated, it's the same point on the tree, just to make that clear for the listeners, right? It's that node on the tree directly parental to SARS-CoV-1 or SARS-CoV-2, where I'm talking about time and Spyros is talking about place. And it's really kind of like fusing those estimates together. Is what we're trying to do in the paper. And I mean, we don't make any assumptions besides the fact that it's a diffusion process, but we do see that our estimates of virus lineage diffusion are actually pretty similar to other people's estimates of bat diffusion. And so even though we don't directly pull in the bat diffusion to inform our inferences,
it's
or
nice
estimates,
to see that it comes out to be similar by different methods,
which, is what we would expect ultimately.
Yeah.
Very cool. All right. Cool. So now the money question is basically, so how do you assume that from these, very constricted areas, the virus made it all the way to Wuhan? Any ideas? In the paper you outlined something.
Let's go for who wants to go? Let's go Spyros then.
I want to hear both anyways. So.
Well, we're going to hear both, don't worry, but
I think there's different. So what we later do in the manuscript is say, okay, we've inferred how fast these viruses diffuse, regardless of in their bat populations and regardless of bat movement, but that diffusion rate matches with the diffusion of the relevant bats, as Jonathan mentioned. And then we say, how likely is it that the
bat flies to Wuhan?
Yeah, that the bat from that parental that where that parental virus was found flies all the way to Wuhan, or flies all the way to I forget the name of the city. So Guangdong province, SARS-CoV-1, right?
Yeah.
In both cases.
So you've got this like dying exponential or something of like probability that it makes it all that way there or something like that.
Mm-hmm. So you either need a really buffed up bat that can play really fast.
The what is he called? Wilson Kipketer of bats or whatever. Yeah, the marathon runner of bats.
Mm-hmm. And at the same time as it does that, is infected with a coronavirus. Or you need to assume some other process, right? There's different. I think what we, based on all the other evidence, what we think is the most likely explanation, and we can discuss more explanations, is animal movement and farming and animal trafficking. And this is because in both cases of SARS-CoV-1 and SARS-CoV-2, there's implications of farmed intermediate animals being involved in the passing over to humans. And there's, if we had any idea of what animal trafficking routes looked like in China, we would have overlaid that and modeled it onto our existing knowledge, but we do not. And a lot of the animals that are infected with these viruses, for example, pangolins, that have made the news multiple times, mainly because it's a weird exotic animal. It's kind of cute. All the two distinct SARS-CoV-2 like viruses that have been sampled in pangolins were found in sick pangolins rescued in pangolin sanctuaries after they were seized in customs while entering China, and they were illegally trafficked from what is probably Malaysia, but again, we do not know where they came from.
Yeah, I would say it's like we would probably also want to have these supply chains in the countries surrounding China as well, right? If we're seeing those pangolins like being taken at the border of China, it means that there were viruses elsewhere. And now we've seen in several papers, right, very similar viruses sampled in Vietnam and Laos and so on, right? And one way we could think about pangolins is it's almost like a satellite for what else is out there, right? Of stuff that is likely circulating in bats or other animals.
Oh, a sponge. Yeah, so I had a conversation with Linfa Wang, and he said, bat surveillance in China is way better. And I mean, it's still not sufficient, but it's way better in China than it is in the rest of Southeast Asia. And if he had $1 billion, he would spend 90% of that looking, outside of China and Southeast China to, look at bats there, look at wildlife traffic routes there to look for the origin. So I think this paper also supports this notion that actually, maybe the wildlife trade has to do with some kind of upward movement from poorer areas towards the richer Chinese markets and cities. Because in the end, pangolins are luxury goods. These are not, cheaply transported animals. There's a lot of money involved in this.
So that raises the question, though: is any of that work being done, following up in those countries, following up those trade routes? Because people will say, look, there's not enough being done, on biosecurity, these kind of things. It's very, very important. But it seems like here we have a lot of information suggesting that this is a very sensible, very plausible route of where this virus might have come from. And yet we don't seem to have much follow-up on that. Jonathan, is anything going on the ground to chase that up? I guess not at the moment. Or
I mean,
we're aware
I think it's a little bit hard to say. Right. Some of the stuff that is probably being done might be challenging to get published.
Yeah.
Some of
but
it,
it's also illegal, right? I mean, it's always hard to follow up on something that's illegal. Simple as that.
Right. Some of it's just like really poorly funded, even though people want it to get done. Looking for a particular, I think, study that was I don't know if it's still pre-printed or not, or if it's just a preprint or if it's published or not. But for instance, there was a paper that, it looks like it was posted in July of 2022 about civet farming practices in Vietnam. And we know civets were involved with the emergence, or at least, related to the emergence of SARS-CoV-1. And they are susceptible to such viruses. And so, people are doing this work. Maybe we'll see more of this work being put out from the countries surrounding China than China itself, but I can't really speak to that. That's, I think, some of the other people who you've interviewed would actually know much better, at least than me.
What is happening, however, is sampling, which is also very important. When we first looked at SARS-CoV-2 origins and compared it to the sampling efforts, quite nothing fancy like we're doing with this paper, but just looked at the regions where SARS-CoV-2 like viruses had been sampled. We spotted Laos as kind of an in between where we've already sampled SARS-CoV-2 like viruses, and people went to sample there and found SARS-CoV-2 like viruses. In our paper, in the preprint that we posted in July, I don't even know what month it is now, but July.
That's what happens when you. That's what happens when you defend your thesis. For us, time just disappears. I understand.
I also managed to go on holidays in between, so that was even worse. But there were no samples from Vietnam, and in between these 4 months that have passed, there have been preprints and
Hassanin and Al, I already having queued for a conversation.
Yeah, absolutely. So the sampling is happening, and I think people like Linfa Wang and many other groups are really trying to get the sampling done, and that is really important. Now, as Jonathan said, when it comes to things that could potentially affect a lot of economical factors, like animal trade, things get politicized, and there's other kind of factors involved, and it becomes much more difficult.
Well, virus hunting has also become politicized. Have you not noticed that the cancellation of Division? How do you feel after doing this study and you're hearing that basically the US, canceled a program that was in 2021 still considered super relevant for pandemic prevention? Now, given your research and that you said it's important, you think it was a good idea to do that?
No, I mean, I am of the belief that I think Spyros and I are both of the belief that we should continue sampling such viruses in bats and also in animals linked to supply chains. It's important to know what's out there. Other researchers might disagree. Our colleagues don't.
And by having funding for this kind of research, you ensure that it's more secure than ever. You ensure that there's more security in terms of equipment. You ensure that there's more security in terms of documenting what's being sampled and whatnot. Funding is key, and I think most people involved in science understand it, but sometimes they tend to pretend they don't understand it.
Yeah, so this is also something that I figured out when I was doing the kind of, the research into this topic a bit. The predict study, it was oversold to a certain extent to say, oh, we can, like a weather report predict, the next pandemic outbreak. But, and of course, this is not always possible given the diversity and all these factors that complicate these issues. And some people are very critical of this. But the important part of the predict program was the training and, the boots on the ground that they created in these countries to do this essential work that now enables, for example, people like you to get a better idea about not only, coronavirus evolution and recombination, but where are these coming from? What do we learn about how pandemics arise? And we created a lot of people, or we used to create, I have to say, a lot of people that are now kind of, working at the forefront of these viruses and are able to sound the alarm when there's a dangerous virus, let's say, breaking out in a village before it reaches the big city, before we are hit by viruses we never heard about that spreads asymptomatically and rolls over the world before anybody knows what's going on. So,
I mean, this work cannot really happen without funding, and better funding will also just allow much safer and better versions of the work, better training, better resources, better institutes, and so on. And as I think is pretty obvious, none of our work would be possible without all of these folks working on the ground, both scientists and researchers, as well as medical professionals. And if we start taking away, this kind of virus discovery and sampling work, then people in our field aren't going to be able to do this work on trying to understand deeper histories and relevant histories for virus emergence as well. And so you're really putting dark spots on many fields as opposed to just finding a virus.
Yeah.
And that's not even getting into the idea of therapeutics and diagnostics.
So this is, I think, a very important aspect that people are not considering, that when you create some artificial stops to research, because research is so collaborative, one piece of the puzzle here enables a lot of other fields to suddenly, fill gaps that we didn't know about before. And if you artificially remove some of these pieces or, sabotage them or outlaw them, also, certain areas of virology are at the danger of being, banned, at least in the US. People don't understand that there are ripple effects to putting artificial barriers. Spyros, you wanted to say something as well?
Yeah, so I mean, I was speaking at a vaccinology conference last week, which is weird for me because I'm nowhere near the field.
Yeah.
But what I was trying to get across to this amazing audience was that for SARS-CoV-2, we had SARS-CoV-1 vaccine programs that there wasn't any actual product, but the research happened. There was MERS-CoV vaccine programs, and all of this made us much better prepared for making vaccines for SARS-CoV-2, which was a fantastic effort.
And got the Nobel Prize today, actually.
Yeah, absolutely. and saved many lives in this pandemic. But knowing the diversity of viruses that are out there is crucial as well. If a virus causes a human pandemic that we haven't seen before, and we haven't had vaccine programs for similar viruses, then all we have in our hands to be prepared is the known diversity of related viruses that is out there.
So we start basically from behind, because the moment a virus spills over into humans, the clock starts ticking, trying to contain it, trying to stop it, and we just throw away our extra preparation time in a certain sense, because we cannot stop these viruses from spilling over. It's going to increase. Climate change is going to drive species movement. We have increased urbanization, deforestation still around the world, new travel infrastructure, people travel faster than ever. So our whole civilization is built on faster transmission for viruses. And if we don't give ourselves some time ahead to prepare for them, we will not be able to manage them, even with the best surveillance programs. Even if we, hit it the moment somebody has a fever in the hospital, the alarm bells ring, doesn't necessarily mean we will be able to do it if we have no advanced preparation time.
I just want to check on people's time, because I know we've done like an hour and 20 minutes. Are people okay for Philip? I don't
10
know
more.
How much?
10 more minutes.
10, 15 minutes.
I have
Is that
10 more
okay?
minutes.
Yeah.
Okay. So now the last one is a bit more personal, because I think
Jonathan wanted to say something there. Actually,
sorry.
just for you.
Oh, no, I was just kind of going to go a little further on Spyros's point regarding like the vaccine developments. There are groups now trying to develop, for instance, a pan-sarbecovirus vaccine, right?
Yes.
We'll see if and when that makes it out. But the better diversity we have for these sarbecoviruses, the better vaccine that we're going to be able to create, and one that will be hopefully more relevant to anything that spills over later, right? And people are doing similar things for influenza. And all of this really requires that type of virus discovery research.
Great. So I think this is a good point to stop with the science and go a bit more into the personal. I know scientists usually dread this part, but we make it, short and painless, hopefully. So
don't have to say anything if you don't want to. Well,
yeah, but can you talk a little bit about what is your experience with the conspiracy theories that were circulating? Have you personally been, targeted for, insults, which is probably the easiest to just, discard, or harassment, or has there been worse experiences? Anything that comes to mind?
You want me to?
Okay, Spyros.
Yeah, I mean, it's been a journey, especially on Twitter, that I
Neither of you engaged that much, really. It's because I think you think it's not really worthwhile, right?
Exactly, and that's the key. And we're both we've both been very busy. I mean,
of course, yeah.
I know that John has
better things to do. Yeah.
And it's honestly a matter of having better things to do. Sometimes I remember we had early on in the pandemic, we all had slightly more time because we were locked in our flats and didn't do anything else. So we had a Slack channel that was feeding into old tweets that like David Robertson that I work with was getting tagged in, and although I had blocked a lot of the people getting engaged in these fights, internet fights, already, I could still, like, when I was bored, scroll through the Slack channel and see all the Twitter fights. But it's really not worth your time at the end of the day. And I think we've tried to do enough kind of public engagement, try to get the science across. But I personally feel like trying to explain things to individual people that may or may not have the best motives behind engaging with me online is honestly a huge waste of my time and my energy. So I generally try to avoid doing that. And I was chatting to Andrew Rasmussen last week, and she said this interesting thing that if you want to be out there, if you want to have a presence and talk about something that is important and gets attention, you will get people engaging with kind of malicious motives, and you just have to be prepared for that. And 4 years of doing this I excuse my language, I don't give a shit anymore. But yeah, that's my take on it.
I mean, me and Philip have, had a couple of percent of what you guys have probably got, and how frustrating and just exhausting it is. It's just absolutely ridiculous. So I mean, I can't even imagine after 4 years of having to put up with this nonsense that people just they just don't want to engage with it.
How about you, Jonathan? Was there an impact on your outreach or how you communicate with the public, your work?
I mean, for me, I think I was a bit more willing to do some outreach earlier in the pandemic. And because of the nature of, just online discourse on platforms like Twitter and elsewhere, I have had decreased bandwidth to continue engaging as consistently. I fully agree with Spyros. Like, I don't think it's always worth the time. Some of our colleagues, like Angie and a few other folks, are, I think, a bit more open to engaging more online, and it's really wonderful that they're able to do so. But also engaging online comes with its difficulties, which a lot of us have faced. And it's really unfortunate, right? Like, these folks have probably very ulterior motives. It's not my responsibility to figure out what those are necessarily. But I would rather focus on the actual research and the science. If there are, opportunities or venues to, let's say, directly engage with like larger audiences or the public, I'm very open to those. But in sort of like a Twitter bubble, that's like less interesting to me.
Yeah, absolutely.
And as like other people that we work with like to say, like Christian, Eddie, and so on, the hope here, and I think we have confidence in this, is that eventually the science will win out. And that's what's going to be remembered historically. Our papers are literally about evolutionary history, right? And we are trying to document what happened in these times. And I hope that like this work is able to just keep doing that.
And I will say that both of you are always incredibly generous with your time and helping both of us to understand what's going on, particularly me, who is not very knowledgeable in the virological field. So you do a great job of doing that and helping me understand the papers and helping Philip as well. So I don't think there's a requirement for you to engage with every single person who turns up to try.
No, absolutely not. My worry is, and I think I've articulated that somewhere else, is that, online chatter you can ignore until you can't, right? Because as we've seen, it cascades sometimes into real life repercussions for people. We've seen, real life threats. People really, Peter Daszak receives white powder letters. He has his FBI agent on the short dial because he gets threats on a regular basis. There are kill lists and death lists of many prominent virologists. Christian Andersen was dragged for a show trial in front of Congress, and others are coming soon. I mean, are you worried about these real life repercussions at some point, or you feel like, it's usually the professors and the PhD students, and we are kind of, away from that. So do you ever consider this when you talk somewhere online?
You don't really want to find yourself worrying about those things in the first place, is my take.
Okay.
It'd be ideal if none of my colleagues, including myself, worried about things or faced these like horrible things in the first place. But here we are. I think both of our involvement has been more on the kind of sidelines, and we definitely don't draw enough attention. At some point, I would, if I found myself seeing that happening, I would definitely kind of step back. And this is not right. People should not step back from saying the truth about the science, because
they're worried about being targeted. They're worried about their jobs. They're worried about their families and so forth.
Yeah. I've seen that Spyros tends to engage when something is just so stupid in the genetics of viruses that he just can't help but jump in. That tends to be your operating procedure from what I've seen, Spyros. You just
It depends on how much.
So silly, I have to get involved here, I'm afraid.
Sometimes I have a bit too much extra time in my hands, or I'm bored of doing any actual work.
No, but it is, and just to reiterate, I think it is important. It is also important that, people like you that, have a certain degree of expertise and are, courageous enough to go and speak up, that you do, because if we just, leave all the conversations to a few people, then their harassment gets a lot worse. And if everybody else is bullied out of the conversation, then, nobody speaks intelligently anymore about these issues. And you have been consistently speaking very intelligently about it. In fact, you have been one of the most successful PhD students that I know in this field. And
although,
I think,
unfortunately, PhDs are not counted by Twitter followers, so that's
exactly. So yeah, I hope, and this is hopefully a bit of a last kind of positive note, is, what's up in store for you? What do you still want to proceed as virology professors in the future? Or you're gonna say, I'm done with virology, I'm gonna do something else with my life? Where do you see yourself in 5 years? And what you want to do with your lives now that you're had a very successful start into this life?
Jonathan, want to go first? Because I think about it.
Well, I'm still a PhD student, so I need to finish that. Yeah. And first, I'll also say, neither Spyros nor I are virologists, just to not offend any of the virologists out there.
Sure.
Maybe Spyros, I did say before.
Okay, never mind. Spyros, you're a virologist. I just do things on my computer and hope it works. I just press buttons all day. And yeah, so after I finish, I will hopefully postdoc for a bit, and then we'll see how that goes. And I will try to stick around a little longer in an academic life, but it's hard to fully know. And I'm happy to continue kind of in the same general field. I think it's been really rewarding to meet folks like Spyros and yourselves, as well as many of our colleagues.
There's worse places than California to keep studying these things, right? If you're gonna continue in California.
I don't think I'll continue in California. I don't really know of better places, weather-wise. So
you should see the rent in California.
Get over to.
I never said it was the best place financially.
Get over to.
They have a nice bioinformatics institute in Barcelona, directly at the beach in the city.
There you go.
Check it out.
That'd be nice.
And it's actually a good institute. It's one of the better ones in Europe.
Or you can swap California for Scotland if you.
Also an option.
There you go.
Spyros, did you think about it?
Less sunny weather up here. Although we've had 17 degrees yesterday, which is warm.
Where are you? Glasgow or Edinburgh? I forget which one.
Glasgow. Yeah. I mean, I think my honest take is that academia is a hellscape. However, having said that, I think we've both found ourselves in quite privileged positions because of the people we happen to work with and the kind of how much we dived into quite big issues that were current, and a lot of that was luck, and a lot of it always is luck. So I initially I'd like to take up the kind of, in a way, privilege I have in the academic world to carry on with academia. But that's not to say that I might just switch jobs completely in 5 years and I don't know, run a coffee shop or whatever.
That's the normal post-PhD reaction.
I'm sure it's not. But yeah, I might look for an escape from academia at some point. Until I figure out that all the adult jobs are actually pretty hard. So
running a coffee shop is also an adult job.
Yeah, exactly. That's what I mean. Yeah. So maybe sticking to academia is the best option. We'll see.
So
and
for
also
me,
with the
it's
hopes that we won't necessarily have much more online, let's say, attacks or abuse in the coming years.
Switch to
No.
plant viruses. That's the other. No
Awesome.
one cares about plant viruses. No, that's
famous last words.
Oh, yeah.
Starts wiping out all the grade supplies or something, and then
it's a real option. And plant viruses are actually really, really fascinating. I should rephrase my last sentence. No grifter usually cares about plant viruses.
Oh, you
No one cares
wait,
about
you wait, you wait. The
potato
natural
blight or tobacco mosaic viruses. Nobody's on that grifter.
Next major outbreak.
Yeah,
across the
wait,
world.
wait for it. The next
Spyros
potato
is on
plague.
it way
Next
too
potato
early.
plague that comes.
Shit. You know, they're gonna when we have the potato virus outbreak, they're gonna like rewind on this and be like,
Oh, of course, yeah.
So yeah, any wish to face more public facing roles in the future, or you're like, you know, I've had enough spotlight for now, and I'm gonna do my work more quietly. Is that the feeling? Or is the feeling like now really we have to, you know, get out into the world as scientists and fight the good fight?
I mean, public engagement, public facing in different ways.
Yeah.
Right. Like, I don't necessarily need it to be through Twitter, but I'm generally speaking funded through the public. And so my research is for them. And so I am interested in engaging with the public insofar that like I want them to understand what I'm doing. And I think it's important for the public to have a good idea of what kind of the landscape of research is generally, so that they also kind of can somewhat inform what questions are interesting. Because it's not just about like what we alone find interesting and important.
Yep, I totally agree with that. And in terms of media presence, ideally less of that would be great.
So this is wonderful. I just want to reiterate because, you know, people have this weird idea that scientists are either in it for the money, which anybody in academia is like, it's ridiculous, or they're in it for, you know, the publicity, the prestige, or the shaping. But I think most of us still, although the incentives are broken when it comes to publishing, although there is, you know, things that are not always ideal and they're cheaters and stuff like that in academia, most people are like the two of you. They're idealistic. They want to work on real problems. They want to work collaboratively with others and really work for the public and try to, you know, help people get a better understanding of our world. And I think this is so important. And thank you very much for being here with us and highlighting this again with your actions way more than our words could. I think this is really important for all to see. Thank you so much.
Yeah, thank you so much for taking the time. We really appreciate it.
Thank you for having us. I mean,
thanks for having us.